Okay, it looks like it's leveled out. We'll go ahead and officially get started. Good morning, good afternoon, and good evening, everybody, wherever you may be. I would like to officially welcome you all to the last installment in Sinobiological's webinar series titled Lock and Key. This is where we explore very cutting-edge immunodetection technologies from a variety of scientists, corporations, and even academic institutions. We have a very special presentation for you today from Dr. Bob Das. Before I introduce Dr. Das, I just want to mention one housekeeping issue, and that is I would like to ask everyone that as you listen to Dr. Das's webinar, that if you have a question, please type that question into the chat box. And at the end of the webinar, I will scroll through those questions and get to as many of them as possible, asking them verbally of Dr. Das, and then he can answer them. Any that we don't get to, we can certainly reach out to you offline and get those answered. So again, type your questions into the chat box and we'll get to them at the close of the seminar. So with that, it is my pleasure and honor to introduce to you our guest speaker today, Dr. Bob Das, who is a senior scientist at the company Sartorius. Dr. Das is a senior scientist on the Octet applications team at Sartorius, and he actually joined the company when it was known as Forte Bio back in 2015. He joined as a field scientist supporting the Octet, BLI, and Pioneer SPR technologies in the Mid-Atlantic and Boston-Cambridge regions of the United States. He has more recently supported customers in the Canada region, and prior to joining Forte Bio, he worked at GE Healthcare in India in the same or similar role, supporting customers on GE's analytical products that included SPR technologies such as BioCore, as well as other proteomics technologies and imaging platforms. Dr. Das has a PhD in microbiology from the University of Agricultural Sciences in Bangalore, India. And the title of Dr. Das's seminar today is Accelerating Discovery and Characterization of Advanced Therapeutics with High-Throughput Biomolecular Interaction Analysis. And with that, Dr. Das, I will stop sharing my screen and turn it over to you.
Thank you, Rob. Thank you for the kind introduction. Very kind of you. Let me share my screen. Okay, I hope I have. This is when I lose my. Wait a minute. Sorry, I just hope I'm getting the right one here. Wait, can you see my screen?
All right.
Right. Thank you so much. Right. Hello, everyone. Again, good morning, good afternoon, good evening. Thank you, Rob, for your kind introduction, and Rob's been very kind for the good words he's used. So, as Rob mentioned, I'll be talking on accelerating discovery and characterization of advanced therapeutics with in a high-throughput fashion using the Octet technology. Again, my presentation itself will have a quick introduction to who we are as a company in Sartorius, and then have a brief introduction to the technology itself, what the Octet is all about, and what's Bio-Layer Interferometry. And I have a few examples of where and how we've used the Octet to help customers in their research and discovery of therapeutic molecules. Right? With that, I would like to welcome all of you all to Sartorius. We would like to kind of believe ourselves to be a partner in life science research and in the biopharmaceutical industry. Our mission is to empower scientists and engineers to simplify and accelerate progress in life science research and bioprocess, enabling the development of more better and affordable therapies and medicines, to people at large. And towards this end, our vision is to bring in experts so that we can help our customers in being able to give better health options to people. As a company, we are about 150 years old now. We started in 1870, way back by a 24-year-old, Florence Sartorius. We started as a weighing balance company in a way, if I could use that. And over the years, we've seen ourselves to grow into becoming expanding our offerings and then being a leader in life science research itself. Right. We have about 60 different locations worldwide. We're headquartered in Göttingen, Germany, about 10,000 odd employees. We are about 2.3 billion in revenue and so on. Again, this is just a quick overview of where we are located across the world and how we are there supporting our customers. Now, as a solution provider itself in the biopharma and life sciences market, probably all of you all have heard as, when Sartorius is there, probably the weighing balance comes to mind, or the pipettes and so on. But we've been pushing to kind of get ourselves into the life sciences research and market. Towards that end, I think 2016 we purchased the IntelliSite, which is a high-throughput flow cytometry-based technique. And expanding on that, in 2017 we acquired IncuSite, which is essentially trying to get an imager within an incubator. And what that allowed customers or researchers is to perform live cell imaging within the environment of an incubator itself. Right? And more recently in 2019, Sartorius acquired Octet platform, where the Octet has been, a market leader in a way in the label-free environment, and essentially trying to expand our footprint in the biologics market and the biologics industry. Right. And then, of course, we also have the Amber systems, which is fermentation, and then expanding on the fermenters, we have different ranges of fermenters from very tiny analytical scale to manufacturing scale fermenters. That's just a brief introduction of who we are. Now quickly going into the seminar itself for the topic for today's discussion. Again, I'm going to read that again. But briefly, what we're trying to kind of do over here is to go, have a walkthrough of what the Octet, the basics of the Octet technology, and then I have a quick couple of slides to show you examples of how we use the Octet in antibody discovery. Right? I'll be focusing a little on talking about more advanced therapeutics, where what we're trying to kind of classify as advanced therapeutics over here is the use of bi- or trispecific antibodies, antibody drug conjugates, nanobodies, and so on. Right. So I'll try to kind of give a show, illustrate with a couple of examples of how it can be useful. Now, again, the Octet itself is an optical tool, and what we're trying to kind of characterize over here is to do something called label-free biomolecular interaction. Right? So why do we see label-free, or why do we say label-free? Sorry, is for the fact that conventionally, when one had to do binding studies, we typically attach a fluorophore or a tag to one of the molecules, and then perform a binding experiment. In a label-free environment, what we're trying to do over here is to be able to have these two molecules interacting without the need for a tag or a fluorophore, and then characterize that interaction to be more realistic in how these two molecules would behave. Now, within this field, this realm of label-free interactions, what can the Octet do, right? So there are a couple of different assays that one can perform, or studies that one can perform on the Octet. Again, broadly, or placing them into 4 different categories, we can do something called binding study specificity or binding specificity. And the idea here is to kind of quickly check, do 2 molecules interact or not? Is there some specificity in the interaction? Expanding on binding specificity, then we would say, okay, well, 2 molecules do interact. Then can you characterize how this interaction progresses, is it a tight binder, a weak binder? How strong is the affinity between these two molecules that have just formed a complex? And then speed of that interaction, right? So we look at what is the on rate of that interaction, what is the off rate of that interaction. Further in the Octet, because of the way the instrument is designed, and I'll quickly try to illustrate what we can do there, is to also be able to perform concentration analysis. So for example, if you had to measure the concentration of, say, IgG samples or of a recombinant protein in culture supernatants, then very quickly we can design an assay where we can use our sensors to dip into a sample and then measure the concentration of the unknown molecule. Again, illustrating that further, where would we typically use some of these different assays or applications? Binding kinetics, for example, the Octet has been used in antigen-antibody characterization, protein-protein, any biomolecular interaction, protein, DNA, protein, lipid, sugar, and so on and so forth, and drug discovery. Steady-state kinetic affinity interactions, again, within the antigen-antibody interaction space, drug discovery space. Sorry, let me just quickly get my, sorry for this, pointer. Sample quantification, say antibody quantification, clone selection, glycan profiling are some of the examples within the quantitation space where the Octet can be used. Epitope binding experiments or cross-competition experiments. The idea over here is if one had a panel of antibodies against an antigen, the idea here is to see if you can map how these antibodies bind to the antigen. Do they bind to the same epitope, or do they have different epitopes that these antibodies bind? And based on that, of course, and the intended purpose of that experiment. So for example, are we looking at trying to design a reagent pair versus are we trying to see, can we come with a synergistic kind of an effect of two antibodies binding to two different epitopes, increasing the efficacy of that treatment. So epitope binding experiments could be used for various purposes. These are some of a very broad characterization of trying to bind these different applications. I have a slide where it kind of expands a little more on these. Now, what enables us to perform our different experiments on the Octet, right? And what we have are something called a dip and read biosensor. We call our biosensors the dip and read biosensors. Typically, what one does is, if you're able to make out from this insert over here, this is how a biosensor looks. It's a fiber optic rod encased in a plastic hub. And just the cartoon view of this would look something like this. The dimension of this fiber optic rod is about 600 microns in diameter. And just at the very tip of the biosensor, we have something called a biocompatible surface that allows us or enables us to form different chemistries and attach different molecules. Typically, one of the molecules or one of the interacting molecules are attached to the surface, and that molecule is called the ligand. And the second molecule is presented within a 96 well or a 384 well plate within these wells, and that molecule is called the analyte. And again, we have different chemistries which enables us to attach different molecules to perform these experiments. Right. And what is it that we measure during an experiment or during or what is it that we detect during a measurement, right? As I mentioned, the Octet is an optical tool or a device. And what we kind of try to measure over here is the change in the interference pattern of a light wave path. Right? So if you could just, at the center of this slide, the insert over here is a depiction or illustration of the tip of the biosensor. And what we're trying to measure is any change that occurs at the tip of this biosensor, right? So if you could imagine this biosensor being dipped into buffer, what we see at the detector is something like a sine wave. Essentially, there's a light that's shined through the biosensor. Part of the light is reflected from what we have, something called a reference layer on the sensor, and the second layer, which is the biomolecular layer that we attach. Right? And what we try to measure is the phase difference in the interference pattern between these two reflected light wave paths. Right? So we look something like this. Right. So as something binds over here, there's a phase shift in that interference pattern of the reflected light. And what we as users see is an upward trending curve, and usually indicative of either something being attached to the biosensor, which is, for example, immobilizing the ligand, or it could also mean that there's an interaction of the analyte, immobilized ligand, right? Now, as the molecule falls off or dissociates, you get something called a dissociative profile, right? So the sine wave goes back to its original position. What we as users see is a curve that trends downwards, typically indicating that the complex is, falls apart. And the slope or the rate at which this occurs gives an indication of how strong is the complex on the surface over there. Now, integrating these two parts of the curve, one can get the association rate or rate at which the complex is formed, dissociation rate, or the stability of the complex in some sense, then get the overall strength of that interaction, right? Bear in mind, the intensity of this signal is dependent on what we call as the optical thickness or density of the molecule that we can build at the tip of the biosensor over here. Now, just a quick insert over here. Typically, this is how the inside of a machine looks like. In this example, we have an 8-channel system. So there are 8 sensors that the instrument can pick, and then progressively dip into different wells in a 96-well plate that's positioned over here. Now, what are the different kind of molecules that one can test on the Octet? I think it's one of those techniques by far which allows us to, or enables our customers to kind of test a wide variety of samples and sample types, right? So it can go from small molecules, typically 150 Daltons and above, all the way to viruses or virus-like particles, lipoparticles, nanoparticles, and so on. So in that dimension. So within these two extremities, we can measure more or less any kind of biomolecule. So it can be, proteins, DNA, protein complexes, and so on. And again, in terms of applications, of course, ELISA, replacement, bioprocessing, biosimilars, and so on. So the idea over here is to give you a sense of where the Octet could be used and for what kind of molecules can be tested on the Octet. And right. Quickly on this slide, sorry, excuse me for that. Quickly on this slide, trying to show you the different offerings that we have within the Octet portfolio, right? So again, the different systems over here vary in their throughput and their sensitivity, going from what we have as a single channel manual system all the way to what we call as the RH96, which is a 96 channel system where one can measure 96 simultaneous measurements if one desired, right? Again, we also have in between these two extremities, we have a 2-channel, a 4-channel, or an 8-channel system. The RH is a 16-channel system. The smaller boxes typically use a 96-well plate. So within the 96-well plate, we have both the reagents and samples contained. The larger boxes, the bigger boxes, the RH16 and RH96, in addition to the 96-well plate, can also accommodate a 384-well plate and a second type of 84-well plate, what we call a tilted well 384 plate, which allows customers to use sample volumes as low as 40 or 50 microliters for their experiments. And so if throughput is something that you're looking for and low sample volumes, the big systems would be something that we look for. Again, we've been about 15 years in the market, and over the many years, we've gathered a lot of experience in terms of the various assays that one can perform, a very strong knowledge base in terms of customer applications, support requirements, service, and so on. So that comes into play, having been there and having worked with customers for many years. We have about 2,500 install bases worldwide, with more than 5,000 publications out there. Now, quickly mention what the technology is and what allows or enables us to perform experiments, which are our biosensors. Now, to kind of further make it easy for our customers, we have a whole bunch of ready-to-use or off-the-shelf biosensors, where typically you could just buy these based on your application or aim of that experiment, and then ready to use, where you don't have to do a lot of stuff. So we have about 18 different biosensors off the shelf for you. Most of them come pre-mobilized with some kind of a molecule on the biosensor. So for example, the HQ or the AMQ, they come pre-mobilized with an anti-human FC antibody or an anti-murine FC antibody. So if your applications is largely, say, human human IgG quantitation or mouse IgG quantitation, then you could these biosensors. We have more in a general fashion. We have protein A based, protein G or protein L based biosensors. So if you're looking at antibody from say different species, it could be say goat or rabbit and so on, then you have a choice of picking one of these. Should your molecule have a tag, for example, a biotin tag or a his tag, then of course we have the nickel NK biosensors or his1K biosensors, which allows or enables us to capture his tag proteins. If you have biotin tag on your protein, then you can use any one of these streptavidin-based biosensors to immobilize or attach these proteins. Now, should you have no tag and still want to immobilize a protein, we also have something called an AR2G, which is an amine reactive biosensor. So using classical EDC and HS chemistry, one can immobilize or attach their proteins onto the surface and then dip them into a sample containing a binding partner. We also have a couple of kits, again, very specific to some applications. We have two kits, for example, to look at contaminants within the bioprocess environment. So if you're using, say, protein A columns to purify a protein or an antibody, then we have what we call as residual protein A kit to measure the concentration of protein A leach within these samples. We have an HTP kit for CHO cells. So if you're using CHO cells to express your recombinant protein, and one of the issues over here is the presence of CHO proteins as contaminants within this within your protein prep. And it's kind of imperative and important that we measure how much of these CHO cells come as contaminants and then find ways to kind of decrease or remove those. So we have a kit for measure CHO protein contaminants. Likewise, we have two kits for sialic acid profiling or screening and mannose glycan screening. So these kits are ready off the shelf. Again, if you're having a panel of antibodies and you would like to say, okay, which of these have high sialic acid or high mannose content versus low. While they're not an absolute measure of the concentration of these sugars, but it gives you a quick way to screen ahead, especially when you kind of have a larger panel of antibodies. You can screen ahead of going, say, to mass spec or one of those techniques where you measure the concentration of glycan. You can narrow down the number of antibodies that you can take on to that step over there. Having seen the different biosensors that we have, on this slide is just a kind of, it's not the most exhaustive list, but giving you a quick summary in a way of the different applications that are possible on the Octet. And all of these applications are currently being used by our customers. Most of these applications were developed by our customers independently. Many times we kind of collaborate with our customers and then expand on some of these applications. So going from ELISA replacement using Octet, for example, to see if you can automate an ELISA process that you already have in-house, screening applications, looking at viruses, VLPs, nanoparticles, particularly within the vaccine space, again, DNA aptamer, RNA aptamer, protein binding within the therapeutic space, small molecule binding, preclinical development, and so on. Right? So again, I believe Sinobiologics will share these slides with you all. Should you have any questions or any particular question with regards to your applications, feel free to reach out to us, and we're happy to help you there. Now, one of the advantages or why is a label-free system important, right? Or why are we looking at the Octet, right? So coming to more specifically within the therapeutic space, where we're trying to discover and develop, say, an antibody therapeutic or even a recombinant protein therapeutic, or trying to identify vaccine candidates, right? So, and one of the things we all kind of agree is especially when we are in the early stages of discovery, is to see, okay, is early failure better, right? Is it good to identify molecules that fail earlier in your screening process, so that all the issues that come up with it later are not there, because it's a very cost-intensive process, taking many years. All of you are aware of that, where it takes many years to develop a therapeutic molecule, and a lot of us now identify that the sooner we can identify a molecule that fails within certain quality attributes, the better it is for the entire process of discovery, right? So that's where we could kind of position and say, okay, this is where the Octet can help us, is to say, okay, you could run an assay probably in your lead selection process itself, trying to use an Octet and say, okay, based on a certain quality attribute, is this molecule better off, or does it stand a better chance to become a successful molecule, rather than, than not, right? And some typical experiments or assays that one, again, we suggest is doing kinetic experiments or looking at immunogenicity, avidity, and so on, depending on the kind of therapeutic molecule that we're discovering or studying, right? Again, kind of expanding the thought on a little bit on a thought with an illustration, right? So classically, we did study kinetic assay, binding kinetics, right? So typically ELISA, for example, would be used to doing a binding study experiment. And what ELISA or even, membrane binding study experiments would give us is the affinity of that interaction, or the K-D of that interaction, right? So if you look at this ISO affinity plot over here, where we have on rate versus off rate kind of plotted over here, these three molecules along this isotherm over here have the same KD value or affinity value. Now, this is what an ELISA or an endpoint assay kind of would give us, right? Now, we kind of understand now that, especially when you're doing drug discovery and designing drug molecules, this information alone is not sufficient. It would be a lot more better to see, okay, well, these three molecules have the same affinity, but how do they actually come out there? How do they actually behave, right? So if we did a real-time experiment, for example, on an Octet, and you'd see that, well, all three have the same affinity, but if you look at the absolute behavior over there, one of them binds really fast, also dissociates very fast. The cyan-colored histogram over here binds slower or associates at a slower rate, but also dissociates at a slower rate, right? So based on these mechanistic behavior, we are at a better position to make a judgment call to say, okay, this is the kind of a therapeutic molecule that I'm looking at, like the black sensogram or the yellow sensogram, or should my therapeutic molecule that I'm discovering or trying to design look more like cyan-colored sensogram, right? And so this is the strength of a label-free system, is to give you in real time what the binding profile or mechanistic profile of that interaction is, right? And the Octet is strong in this, and which is why a lot of our customers find value or see value in having a label-free system. Now, how does one typically set up a kinetic an assay on the Octet, right? It's titled as kinetic assay, but I would say this kind of works even if you're setting up a quantitation assay or an epitope binding assay. The idea is to give you an illustration or kind of in the sense, a cartoon illustration of how a typical assay is set up on an Octet, and to kind of give you an idea of how easy or simple it could be in terms of the setup and analysis, right? In this particular example, it's just a small example to show you a single concentration affinity screen. So, for example, if you're doing, lead ID of your antibody panel, a lot of us typically also do cell-based assays to identify antibodies, but increasingly we're recognizing that, okay, could we, in addition to cell-based assays, if we do binding studies as well, now will that give us more information, right? So will it, put us in a better position to make a better judgment of which of these molecules are better off going forward, right? In this example, we're using a streptavidin-based biosensor. We are trying to immobilize or attach a biotilated antigen molecule onto the streptavidin biosensor. And we're screening a bunch of antibodies over here, right? So in the fourth column over here, you have different antibody molecules at fixed concentrations or at a single concentration. When we typically run an experiment, this is what would happen. These 8 biosensors. So in this illustration, we're using an 8-channel system, so which is why you see these 8 biosensors. Typically, these 8 biosensors would dip into a column of buffer in your 96-well plate, establish a baseline, then quickly move to the next column where you attach the biotilated protein, and then you go into a third column which contains buffer, do like a washing step, and then dip into your fourth column, which is a bunch of your antibodies. And then you come back to a column that contains buffer, measure the dissociation of that bound antibody. Now, if we were to do a kinetic screen or affinity screen, very quickly, you can say, okay, no, this molecule has a tighter affinity versus some molecule has a lower affinity. And based on this information as well, in addition to your cell-based assays, our judgment about a particular therapeutic molecule in terms of taking it forward is going to be that much stronger, right? And again, depending on on the throughput of the system that we're using, how the speed at which an experiment like this can be performed is fairly phenomenal on the Octet, right? So this experiment, for example, could be completed in about 15 or 20 minutes, of course, depending on how long the dissociation rates are, dissociation times are. But something as easy as this could be designed on an Octet. Again, whether we're doing a concentration assay or an epitope binding assay, it's an expansion of this kind of layout, layout that is used. Now, again, a core strength that I would like to highlight the Octet is now, because it's a dip and read format, where the biosensors essentially are moved to your sample, it offers a certain advantages, right? So unlike label-free systems where we use microfluidics, where the sample is taken to a detection chamber or detector or a sensor, in the Octet, the sensor is taken to where the sample is, and that kind of, because the Octet is a non-microfluidics-based system, it offers certain advantages of being able to work with crude samples, and how one can, say, for example, do rapid assay optimization and so on. So if you could imagine, if you could kind of imagine, in theory, if you had a 96-well plate, buffer a couple of wells where columns were used for buffer and so on. For all your testing molecules, we could, in theory, have 96 different conditions that can be tested on the Octet, right? So you could have antibody 1 in buffer A, buffer B, buffer C, and see how they bind, concentration of detergents, coupling conditions, and so on, right? So a whole bunch of assay optimization assays could be done very efficiently on the Octet. Now, coming more to its applications within the drug research and process development space, I mean, slowly again, kind of segueing into the use of the Octet in advanced therapeutics discovery, right? So if this illustration kind of quickly gives you, we've tried to kind of color code where the Octet applications have been. Again, one is not limited by what we've shown over here. In some sense, if we have a chemistry to attach one of the molecules to the sensor, and if there's some specificity in a second molecule binding to it, I think in all purposes we can design an Octet assay, right, or an immunodetection assay on the Octet. So within the target ID and validation space, the Octet has been used more from doing kinetic applications, is looking at mechanism of action and molecular interactions, ELISA assay development within the space. Lead screening and lead selection, again, we can have either a kinetics-based application or even a quantitation-based application of the Octet in this space. Right? So for example, you can screen for binders in hybridoma, phage, and lysates. Where we're looking at the titer values of the concentration of these molecules. We can also do affinity screening or on-off rate screenings to help identify clones or help identify the appropriate the molecule, right? Likewise, again, mammalian holds clone scaling up and so on and so forth. Lead optimization, process development, QC and manufacture, and you'll see that a whole bunch of applications where the Octet fits in very well to enable our customers do these applications. Excuse me. Now, if you look at the FDA side, when you're looking at the drug discovery workflow, broadly grouped into 4: target, ID, hit discovery, hit to lead, lead to candidate, right? And again, this is known to all of us over here that it takes a fairly long time for one to come up with a successful molecule. Right? And this is just an illustration of a typical drug discovery sequence selection workflow, where we start with a very large number of, say, for example, an antibody library, progressively screening them and then reducing their numbers to something that, something closer to, say, 10 or less, where we say, okay, of 1,000, these 10 molecules show the highest potential, in terms of its efficacy as a drug molecule and in it kind of being developed into a successful drug molecule, right? So a typical funnel that all of us have come to know now. And these are some of the assays that one typically performs in this screening application, right? And if you take a closer look of some of these applications that are listed over here, we can realize that the Octet can be positioned in this entire space over here, where one can do potency assays, mode of binding studies, all the way to stability studies on the Octet, right? So that was just to illustrate, give an illustration or kind of set the tone for what's going to come next. Now, having understood that, a lead screening process typically takes, where we kind of reduce the numbers, starting from a very high number of antibodies or potential drug molecules, right? So now looking at bispecific antibodies or antibody drug conjugates and antibodies, more specifically as examples within advanced therapeutics, right? Now, again, just to introduce why, again, many of us know all of these, but why do we need an advanced therapeutic, right? Or like with any molecule, there are always some limitations, or probably one molecule does not, or one type of drug molecule does not satisfy a whole bunch of requirements. So some of the challenges have been, say, multifactorial nature of diseases in terms of its complexity, drug delivery, efficacy, and so on, right? So these are some of the challenges that have been there within the biologic space. And so we're finding different ways to come up with various combinations of strategies to kind of circumvent some of these limitations and to offer better drug molecules to our patients. So to that effect, I've just say bispecific antibodies, ADCs, and nanobodies as examples to illustrate how Octet has been helping some of our customers in this space, right? Again, a lot of these molecules, their common disease areas have been cancer, inflammatory disease, and so on. We've had about 2 bispecific antibodies out there in the market, or 85 of them in clinical development. There are 9 ADCs approved by FDA as of 2020, about 80 or over 80 being investigated or evaluated, right? So a lot of effort has been put in this space in being able to develop some of these molecules, right? And when you look at the FDA guidance for these molecules, right? So what does the FDA expect when you're designing these drugs and characterizing these drugs is one is to show antigen specificity, to look at their affinity on and off rates, the avidity of that interaction, especially if you're looking at bispecific or trispecific molecules, potency, process impurities, and so on, right? So a half-life of the molecule within the physiological environment, right? So these are quality attributes that the FDA guidance kind of lists us. These are some conventional methods, current methods that have been used to some of these, right? So binding assays, ELISAs, for example, SPR, and so on. But while a lot of these, of course, have its own strengths, but have their limitations as well. And typically has been the difficulty in being able to set up experiments, time consuming, and how these experiments are done, and so on, right? And so we would like to kind of show how the BLI or the Octet Bio-Layer Interferometry technology can help kind of speed in some of these processes, right? Now, again, just a brief introduction or pictorial kind of illustration of a bispecific or a trispecific antibody. Again, I will not get into a lot of these details, but just to kind of showcase that, well, this is a conventional IgG. In a bispecific or trispecific, we typically have a variable region designed in which can specifically identify or recognize or bind to 2 or 3 different antigen molecules, and thereby increasing the efficacy or the way the drug can be delivered. Right. Now, as an example, if one were to design a bispecific molecule, a bispecific antibody, an example of how the octet can be used to characterize this molecule, is kind of is being illustrated over here. A fairly simple example over here. So we have two arms of known antibodies to the HIV virus. These have been designed in as a bispecific antibody using a common human FC backbone, right? So in this example, we would immobilize the antigen, which is the CD4BS ligand molecule. So we use, I think, streptavidin biosensor in this case to attach the antigen. And a classical example would look, an assay would look something like this, right? So you dip the biosensor, attach your biochelated antigen onto the molecule, do a quick wash step, and then very specifically, you can dip the biosensor either into just the parental antibody, which is just a monospecific antibody, if I may use the word, and your bi or trispecific antibody, right? And in sequence, we kind of go dip them into these different samples to show that, for example, in the red sensogram over here, where you show that the antigen binds, or one arm of the antibody binds to the antigen on the sensor surface. Second arm, specific to a second analyte, in this case V1, V2 analyte, also shows binding, right? So it's a classical sandwich kind of an assay where you can quickly show that your bispecific antibody binds to different antigens effectively. And further, one can go on to characterize its binding profile just to the parental IgG molecule, as well as its bispecific designed molecule, and show that, okay, is there any differences in terms of its affinity profile? Is one better than the other? Or just designing a bispecific or a trispecific molecule, does it reduce the affinity and so on, right? So a lot of these mechanistic as well as mechanistic informations can be kind of derived from these studies, right? And then from that, of course, you can expand to see what its broad neutralization potential is in terms of a therapeutic molecule. A second example within the bispecific antibody. For bispecific antibodies, I've just picked up this paper, which talks about this trying to design neutralizing antibodies for Ebola virus, and the idea over here was to come up with a bispecific antibody, which would one of the arms would bind to the Ebola virus, and the second arm would bind to a specific receptor within the late endosome, thereby improving the efficacy of the degradation of the Ebola virus or the killing of the Ebola virus. Right? So the paper goes on to show that the efficacy of having a design, for example, what the authors call a dual variable domain, where one of the arms bind to say the glycoprotein on the virus, and the second arm binds to the receptor binding site in the endosomes, and then specifically targets the Ebola virus within the endosome, improving the drug efficacy. Right? And a simple octet assay could be something like this, where we first go on to show the binding of the antibody, right? Just the monovalent of the conventional antibody to either the Ebola virus GP or the receptor domain, and then further perform an experiment, which is almost like the sandwich assay that we saw earlier, where we first look at the antibody binding to the GP domain, and then the antibody or the second arm of the antibody binding to receptor binding site domain of the endosomes, right? And so the octet can be used to kind of quickly evaluate your design to show that, well, one, there is no reduction in the affinity in this particular design, and or depending on what you're hoping to do, right? So based on this, okay, could I then attach an FC domain, to increase its half-life? As an example, like we should have that as well, whereby we want to say, okay, I have domains, but by designing in or engineering my FC domain, can I increase the half-life of the antibody in the circulating environment, right? So these are just two quick examples of a bispecific antibody. I have one for anti-drug conjugates. Again, not going too much into what an antibody-drug conjugate looks like, what are its functions, and so on. Again, a lot of this information is available. But again, the idea, something very similar to bispecific antibodies, is in this case, we have a conjugate, which could be a drug molecule. We're using the specificity of the antibody in a way to kind of take the drug molecule closer to its site of function, right? So for example, in carcinomas or tumors, you're going to use antibody specific, for example, to a certain surface marker on a tumor, and then the drug molecule, once it's closer in the vicinity of the tumor, the drug molecule then kicks in and bringing about more efficient killing of the tumor, right? Again, when designing your ADC molecules, typically one looks at, okay, what is the half-life of these molecules, because that is one of the things that has that have been kind of limiting is to see if there's an increased half-life of these molecules, and then of course you'll do more functional assays to look at its killing efficacy, right? So in this paper, again, chose this paper to give a quick comparative illustration of the strength of the Octet over here, right? So one can perform classical antibody drug binding, right? So if you're looking at a receptor binding, you want the antibody to go to the receptor and bind there. You can quickly design a screening experiment to show, okay, which of these antibodies would bind at a high affinity to the receptor on the molecule. So for example, for discussion, if I could use, this antibody binds fast, but also has a very fast dissociative profile. Now, is this a characteristic that I'm looking for, or would I look for an antibody that binds really well but stays on the receptor for a longer time? Because these two would have implications on the dosage of the drug molecule, right? So if there is one that stays for a longer time, probably we need a lower dose of it, or fewer times that one would have to take that drug, versus if something that binds well but also comes off, meaning one would need to take more often of that drug molecule there, right? So again, in this paper, the authors discuss using well-characterized trastuzumab variants, both looking at conjugated and unconjugated antibodies binding to HER2 protein, and then further kind of using the Octet to do the FcR and binding assays, right? So again, to say, okay, with each of these conjugates, what happens to the half-life of these antibodies when they're injected in their test samples, right? So being able to perform an FcRn or Fc gamma kind of an assay, in addition to classical binding studies, again expands the kind of information available for us to kind of make a decision as to which molecules to choose, or which would have a better potential as a therapeutic over here. Right. Again, the third molecule over here that we want to discuss with nanobodies. Again, these are just variants. If you kind of look at all three examples so far, they've been typically variants of the of antibodies. Nanobodies, again, like all of us know, are smaller. Classically was identified in camelids. And some of its desirable properties have been small size, stability, strong antigen anti-binding affinity. Limitation, of course, has been lower half-life because some of them lack FC domains. And so, again, current strategies have been to see can we kind of design an FC domain to an antibody to engineer and increase its half-life in the environment. Again, nanobodies have been used more both for detection or detection systems or diagnostic systems, as well as therapeutic molecules. Right. Now, quickly, in the interest of time, I will kind of show a couple more examples where the Octet has been used. So again, this slide is kind of, if I could use, if you could summarize what can be done in the Octet is to being able to do measurements, look at vaccine, therapeutic potency or stability, assess epitope diversity and coverage, looking at single concentration kinetic screens or full kinetic screens to identify mechanism of action and so on, and looking at, say, effect of neutralizing antibodies and so on. Finally, when you have a drug molecule or inhibitor right? An example of a typical titer assay is being able to use, say, our protein, off-the-shelf protein or protein G biosensors, and then measure the concentration of the titer from your hybridoma supernators, right? And kind of show that the concentrations can be as low as 0.5 microgram per mil, all the way to about 500 or 1,000 micrograms per mil in terms of what can be measured. Right? A very good example of where the 16-channel system was used in this example or in this publication, where they looked at selecting high-affinity anti-drug antibodies as a way to design detection molecule or detection reagents, right? So again, anti-drug antibodies are a major concern when we design protein-based therapeutics, and so being able to design an experiment or an assay to measure the concentration of anti-drug antibodies is important. So in this paper, what they've done is to use the 16-channel Octet system, where they're doing something called an Offrate Screening. Many times when you work with lysates, since we don't know the concentration of the absolute molecule present there, which means that we can't do a full kinetic experiment, we can do something called off-rate screening. So as all of us know, the dissociation rate or the off-rate is a concentration independent parameter. So in the absence of that information of the concentration of the analyte molecule, we could still perform what is called an off-rate interaction or off-rate measurement. And then based on that number, we can then group these molecules to say, okay, this is the kind of molecule that I'm looking for or not, and so on. So in this particular paper, the authors have used cetuximab and trastuzumab to kind of identify Fab antibodies expressed in E. coli, right, lysate. So again, the strength of the Octet over here is being able to use crude lysates, right? You don't have to purify these. You could use lysates, bacterial lysates or supernatants to quickly screen these molecules to be able to identify an ideal candidate. Coming to the other application that a lot of our customers in the biologic space use the Octet for is epitope binding, again, also called as cross-competition assay. In a way to kind of describe this is to identify, if we had a panel of antibodies, if these antibodies have the same epitope or do they have different epitopes. In a way to assess the epitope diversity, you can use the Octet. Again, there are various formats that the Octet allows, and it's probably one of the only techniques or the very few techniques out there that allows or gives our users the freedom to have any of these formats when they design the experiment. And again, the choice of each of these formats will depend on the nature of the antigen, either monomeric, or multimeric, oligomeric, and so on. And so having that advantage to use different formats is crucial over there. Again, just an example to kind of show how epitope binding can be used. In this case, looking at a protein from Klebsiella, then trying to identify antibodies, see if they bind to the same epitope or not, right? So in this case, the MRKA antigen was immobilized on a streptavidin biosensor. And then what we do typically is to end up the biosensor into the first antibody, or what we call as a saturating antibody, and then ensure this antibody occupies all epitopes on the antigen, followed by which we dip into a competing antibody or a second antibody, and then see if this antibody binds or not. Fairly straightforward in that sense. So if you look at clone 1 and clone 2, clone 1, for example, here shows binding, which means that it has a distinct epitope, which is very different from the first antibody on the surface. While clone 4, for example, which is very close to your buffer, which almost means there's no binding, probably has a similar or overlapping epitope. So clone 4 and this first antibody bind to the same epitope, right? Again, this is just a very small matrix that is shown over here. On the Octet, one can, again, depending on the throughput or the system that you're using, one could design something like a 64 or an 88 by 88 kind of a epitope binding panel, right? So that's possible. But the idea over here is to show, illustrate that the strength of the Octet and being able to perform some of these experiments. Similar experiment can also be done to look at epitope scaffolding. Again, in this case, is to show that, okay, if we have more than one kind of an epitope designed onto a scaffold, and then screen your antibodies or look at antibodies that bind more efficiently to these, right? Looking at conformation and effect of conformation of a receptor, right, and how that might impact or affect the design of a therapeutic. And again, in this case, looking at the ACE from COVID-19, it's kind of again known now that this particular domain over here, there's an up and down conformation, of the AC protein receptor molecule. So depending on how the antibody binds, does it bind to the closed domain, or does it require the receptor to be open before it binds, right? So the Octet was kind of used in this case to show that, right? So again, very quickly, the green sensogram just shows the spike protein with this up position. So you see a good binding of the antibody, versus when it is in the trimeric form, which is in the closed form, then you see there is low or no binding over there. So using the Octet to look at conformational changes that affect accessibility and binding, right? So gives an important strength in being able to characterize molecules. Then again, looking, once you have an antibody, do we say, okay, do they bind to the same or recognize the same site on the AC, or do they have different sites on the AC? Again, a classical epitope binding approach can be done where sequentially you pass the first, in this case, you put the receptor binding domain on the surface, come with the first antibody, and then come with, say, AC and see how they bind over here, or see if this antibody blocks that interaction of AC to the receptor binding domain, or vice versa. Again, expanding that is to perform a full kinetic experiment to look at neutralizing antibodies, or to show the potential of neutralizing antibodies, and the effect of domains, the subdomains within this characterization of neutralizing antibodies, right? So if you could quickly look at the table over here, the SARS ecto domain, for example, has a high affinity as compared to the whole protein, right? And so in this particular paper, the authors go on to show that probably the ecto domain had an impact, had an effect and in the neutralization potential or neutralizing potential of these antibodies to the SARS-CoV versus SARS molecule. To kind of summarize now, so what we've shown in this presentation is to show the utility of the Octet platform for a wide range of applications in drug discovery and research. And similar principles can be applied to other types of therapeutic development areas, right? Such as small molecules, peptides, DNA, RNA, aptamer, vaccine research, and so on, and where the Octet can be used. Again, key advantages of the Octet. Again, we're going to share the slides, but kind of quickly to kind of highlight some key features is the ability to analyze unmodified molecules, right? Meaning you don't have to have a tag or fluorophore and so on. Being able to analyze in crude samples without having the need to purify samples, so which is very crucial, especially when you're doing screening experiments and so on, very early in your analytics, where you don't have, or you want to purify samples, right? So and so there the Octet has an important role to play there. Being able to optimize assay conditions. It's a non-microfluidics-based system. So the advantages of blockages and so on and so forth are not there on the Octet, right? From an instrumentation point of view, very easy to use. No cleaning required between experiments. Very low sample loading time and so on. Minimal maintenance. And the scalable throughput, right? So depending on the system, you can scale from one of our low throughput systems all the way to the high throughput system, right? Again, why the Octet? Again, a time-saving all-in-one platform in a way to kind of look at your therapeutic investigation. Fast and flexible platform for antibody discovery, epitope characterization, and coverage determination. Versatile solution both in upstream and downstream vaccine development discovery as well. and of course, intuitive and easy to use data analysis software and tools. So with that, I would like to say thank you. If you have any questions, I'll be happy to take them. Again, I have my mail ID over here, as well as Sartorius's website. Should you have more questions, feel free to reach out to us. We'll be happy to kind of help you in any way. Thank you so much again for your attention. We'll be happy to take questions.
Hello, Dr. Das. So this is Jen. Because we're a little over time, so Rob had another meeting, so I will take over. So
I'm sorry about that.
It's okay. It's okay. I'm here, so I will take over the Q&A session. We have quite a few questions here. I will just read through to you. The first question is from Mano Banasia. What is the difference between the Octet technology and SPR? And is the Octet technology suitable to determine binding affinity of small molecules to proteins?
Right. Thank you so much for the question. Well, at the core of it, both the Octet technology and SPR, both are optical devices or optical tools. But the difference is what property of light do we use, right? So in the Octet, we're looking at measuring change in the interference pattern of light as it's being reflected between two surface layers, right? So the biosensor as well as something that's bound to the biosensor. In SPR, we typically look at something called an SPR angle or change in the angle of a reflected light path, which typically is a phenomenon because of something called surface plasmon. And so any change in mass on the surface of the biosensor in SPR affects the change in the angle of that reflected light. So those are the two differences at the very heart of these two technologies. Is though both are optical devices, it's the principle that is different. One is interference pattern, which is in BLI or the Octet. The other is reflected light or total internal reflection due to an effect of surface plasma, right? So that's the two difference. And being able to measure affinities of small molecules, yes, we can use the Octet to look at small molecule measurements. All our automated systems, again, if you look at, we have one manual system, which has lower sensitivity, but all the other automated systems have the sensitivity to look at molecules as small as 150 Daltons. So if you had small molecules, 150 Dalton and above, we can look at that interaction to protein and we can measure that.
Okay, thank you.
Sure.
The other question is from Joseph Arazeli. So it is a similar question to the previous one. So
Sure.
What is the range of molecule and protein ratio that be accurately measured?
Right. Yeah. So it's kind of it's a very important question, but also maybe a little difficult to answer as well, with a single number. But typically something like, say, looking at 1:100 kind of ratio is what we look at. So for example, if you had a small molecule, which is about 200 or 250 or 300 Daltons, typically the size of the protein on the sensor, we recommend to have something like, say, less than 80 kilodaltons, right? So maximum 100 kilodaltons. But any molecule larger than 100 kilodaltons, one would have a bit of a challenge over there to look at small molecule interactions. But having said that, and so that's where the subjectivity comes is, we've had tested molecules larger than 100 kilodaltons and still can show binding. But I would say if your molecule is larger than 100 kilodaltons, it can be a bit of a challenge over there.
Okay, thank you.
Sure, please.
The next question is from Felix Viga. Why does the experiments needs to be run at 30 Celsius degree?
Thank you for that question. So again, we are not limited to run it at 30. It's just that, when the Octet was designed, probably in its initial days, it defaults to 30. But on our system, we have two platforms. So the smaller boxes that I showed, one can go from 15 degrees Celsius to 40 degrees Celsius on those machines. That's the temperature range. So you can pick any temperature within that range over there. On those two big boxes that we have, the current working range is 4 degrees above ambient, all the way to 40 degrees. So if your room temperature is 20, typically you can do an experiment at 25, go all the way to 40. So we are not limited to 30. It's just that the system, as the software design, was defaulting to 30, but you have the choice to change.
Got it.
Sure.
The next question is from Fernando Barrasso.
Sure.
Can you control the pH in these experiments?
So again, the pH itself is something. If you look at there's one small insert that I had very early in the slide, which showed the inside of the plate. Our samples are contained in a 96 well plate or a 384 well plate. So one of the advantages is after the experiment is done, of course, the samples are not consumed during an experiment, so you can reuse the sample. But to answer the question on pH, the pH is something that the user or the scientist would have to do a priori, right? So when you're preparing your samples, you test the pH or choose a particular pH and then dispense the sample into the well. But within the experiment itself, we cannot change the pH. For example, when people do FCR and binding, what they do is they would have, say the antibody at neutral pH and the same antibody at pH 6, and then see it's binding to the FcRn receptor, right? So that can be done, but the control of the pH happens outside of the system, not really within the system or inside the system. Sorry.
Yeah, thank you.
Sure.
The next question is from Chao Chen. So, how sensor type could affect antibody and specific binding? For example, AHQ versus AR2G. Is it related to sensor material difference?
Good question again. Thank you for that. So in both of these techniques, if you say, so the AHQ, so for doing kinetic experiments, we have, instead of AHQ, which we recommend for quantitation, we have AHC, which is recommended for kinetic experiments, but your question is still valid, right? So when we choose a biosensor that has an antibody on the surface, so the AHC, for example, has an anti-FC capture, anti-FC antibody, which binds to the FC portion of the antibody. There is some directionality in the way the molecule is bound, right? So the antibody is bound by the FC domain to the sensor surface, and so your antigen binding domain is exposed over there. Versus if you look at AR2G is an amine-based reaction, so it's a random process. So any lysine on the antibody potentially could be used and it can go and bind to the sensor surface. So one of the issues that one typically sees with amine reactive chemistry, typically where it's a random process, is many times when it binds to the sensor, it can mask the binding site. Right, so when it masks the binding site when it goes to on the sensor surface, then that can potentially come as a false negative, where it will say your molecule did not bind to the ligand on the sensor, right? And that's purely because the binding site was masked. So again, as assay design, we always recommend have a positive control to ensure that the surface is working fine, right? Versus this might not be an issue when you're using an AHQ because you clearly are binding by an FC, exposing the antigen binding sites. So, that design would be better in some sense. But again, now is one better than the other? It depends on what's available to you as a user. If you have the choice of using AHC, you could test that. If you have no tags, for example, then sometimes AR2G becomes the only route to go.
Okay, thank you.
Sure.
The next question is from Samir Acharya. So is this system sensitive to sequential interactions with more than one ligand? If so, to what level can such interactions be determined?
It is. I mean, if you look at our epitope binding assay, for example, in some sense, it is sequential where, you have the antigen on the surface, then you go into the first antibody followed by a second antibody, right? There's some experiments that we've done with customers where we actually worked with larger systems as well. So we've taken nanoparticles, immobilized one layer of nanoparticles, come with a second layer of nanoparticles and third layer of nanoparticles, right? So and we've gone as far as something like 250 nanometers or I think 300 nanometers, from the surface of the sensor without any loss in signal, right? So So again, an important question, especially when you're looking at SPR, because SPR has a decaying signal. So when you have, as you go away from the sensor, there'll be some loss in sensitivity of the signal. But on the Octet, we've not had that issue within our typical working range. Like I said, we've gone up to 300 or 400 nanometer away from the sensor, and we can still do some really good binding studies. I hope that answers the question, but if you have more questions, I can always answer that later.
Okay, thank you. Due to the time restrictions, we still have quite a few questions in there.
Sure, please.
But I will pick the most important ones here.
Sure, please.
So there's another question from Miguel Aloya. So in terms of the analysis of affinity measurements of IgG1 monoclonal antibodies, which model should be used, the 1-to-1 model or the 1-to-2 model?
Good question. Again, a lot of times the application of a model is a user-defined input in the software, right? So while the software offers a choice of 4 different models, the easiest in a way, or let me see if I can. So describing a 1-to-1 model would be the most easiest, right? So it's the most stringent, but also probably the easiest, right? Versus if we choose a 1-to-2 model, then we'll have to give a rationale to say why we chose a 1-to-2 model, right? But the decision to apply a certain model lies with the user, or lies with the scientists. And so now if you're looking at an antigen and antibody interaction, it depends on how we've designed the assay. So if we have the antigen on the sensor and then come with an antibody, then you're typically coming with a monovalent antigen and a bivalent antibody. So a 1-to-2 model in theory would apply. But if you're reversing the orientation where you have the antibody on the sensor and then coming with the antigen, then in theory, each arm of the antibody binds to a single antigen molecule. So a 1-to-1 model would apply. But when designing an experiment, we would recommend as much as you can, if that flexibility is there within the system you're working, try to go with a 1-to-1 model, because that's easier to describe than if you're choosing a 1-to-2 or a 2-to-1 model.
Thank you.
Sure, please.
The next one is from Ansgar Brokel. Is the system applicable to handle gold conjugated proteins, for example, during development of lateral flow assays? And what needs to be considered when working with large nanoparticles in comparison to small molecules or proteins? Right.
To your question, yes, it is compatible. Even if you're working with good GOAT antibody conjugates. Of course, the choice of the biosensor, will be limited to using either protein A, protein G, or protein L biosensors. But if you are able to biotinate that molecule, then of course it expands the choice of biosensors you have. You can then go to structavidin-based biosensors and so on. With regards to nanoparticles, with small molecules, the requirement is to have a sufficiently high density of protein on the sensor, so that when you have these small molecules binding, the small signal is then becomes large enough that you're able to measure it on the system. Again, an ideal system would be to have the small molecule in the sensor and then come with your protein binding to it. But most often, derivatizing a small molecule can affect its binding property itself. It can sometimes mask the binding site and so on. So what people most often do, or users most often do, is to immobilize the protein and then come with a small molecule, right? So the challenge over here is to have sufficiently high density of protein. Versus when you're working with very large structures, say with with nanoparticles and so on, then depending on, are we immobilizing the nanoparticle, or is the nanoparticle going to come as the analyte, then steric hindrance would be a challenge, right? So if you have very high density of an antigen, and you have nanoparticle or high density of antibody, maybe nanoparticle expressing your antigen comes in there, then you could have steric hindrance, avidity, and so on and so forth, right? So those are some of the challenges that you would have with larger structures. So designing or optimizing your sensor conditions or surface condition would be very important when you're working with large structures. So typically avidity kind of effect or even steric hindrance would be an issue there.
Okay, thank you.
Sure.
I will just choose 2 more questions.
Sure.
The first one is from Mano Banasia. So what is the price range of these instruments? For example, the R4 series versus the N1?
Oh, and so I would be the wrong person to give you a number there because in a way, I'm not, probably it would be best to kind of reach out to your local sales managers. And again, the pricing differs across different regions. In the US, for example, it's a different pricing versus Europe versus Asia. The pricing could be very different. But if just to give a comparative, note say so the N1 would definitely be much cheaper than the R4. So the N1 probably could be, I don't know, $20,000-$30,000 US. The R4 could be somewhere in the hundreds or 200, somewhere there. Again, I think so you shouldn't quote me on the price. The best would be is to reach out to our sales managers. But again, if there's an interest to get any kind of a quote or some pricing information, please let us know. I can direct it to the respective sales managers, and they can get in touch with you.
Okay, thank you.
Sure, please.
The very last one from Mark Kamanzine.
Sure,
do you ever use Octet for saliva analysis, such as for COVID antigens? Does variable metrics or of saliva affect reliability?
Right. That's again a good question, interesting question. We've had a lot of, I think, in the initial stages of when kind of COVID came in there, we had a lot of questions regarding this, being able to use saliva. We had some questions from companies working in the airport to see, okay, could the Octet be used as a screening tool in the airport, where they can design assays based on saliva. Now, with regards to the matrix, again, like I said, like we discussed earlier, because the Octet is a dip and read assay where you take a sensor, which is just a solid glass rod, there's no microfluidics, you dip into your sample. It allows us to work with crude samples a lot better than, say, a platform like SPR. And now the matrix composition, of course, will have an effect on the sensitivity of the assay or the robustness of the assay, and that's something that has to be evaluated. But to give you an idea, we have a lot of customers who use serum samples, who use culture supernatant, bacterial lysates, and so on, and have very successfully designed assays on the Octet. And I don't think so it should be an issue with saliva as well. Now, what might be an issue with saliva is, of course, one is the quantity, and then, of course, the concentration of the virus, right? So the kind of assay that we would have to design is something that we'll have to kind of optimize and evaluate. But purely to test it, I don't see a reason why we should not or why we can't. I think we should be able to do it on the Octet.
Okay, yeah, thank you, Dr. Das. This is a really great and interesting talk, and we received a lot of questions, and we we don't have enough to answer them all. So for all our audiences, if your questions has not been answered yet, you could directly contact with Dr. Das, or you can send your questions to us. We can forward you. So thank you for all your time here. We really have a great time here. Thank you for Dr. Das again and our audience. So we are getting close to the end of the webinar today, and I hope everyone enjoy the rest of the day. Thank you. Bye-bye.
Yeah. Thank you, Jen, and thank you, everyone. I would like to thank Biological for this opportunity. Again, thanks for all the audience and participants. Hope you got something out of this. Have a good day, all of y'all. Take care. Stay safe.
Yeah, thank you. Bye-bye.